Thank you for having me. And just to the last speaker, I actually found that talk exceedingly interesting. My own story is probably very similar in that I became an entrepreneur really after my father was passing away in 2015. He was dying from prostate cancer. And I just thought this is an opportunity to take all the experience that I have and utilize it within the industry to train other people, because I realized there was a niche market and there was not a lot of people trained in clinical trials. I found it by accident, as with many of my colleagues, how to do trials, but how to do trials well is so important. And that's why I started my business in training and consultancy. So thank you to the last speaker. I think it's very good to have speakers who can provide ways that people could start their own career, and don't be daunted by it. It comes together eventually. So I consult out my services, as I mentioned, as a consultant to different companies, and more recently I've been working in the area of gene therapy, and this is the reason why I am writing about this area today. It's a really interesting part of the industry, and really, from my view, it really is a game changer in how we run clinical trials and what kind of areas, the therapeutic areas we can treat. So I'm going to start. Let me know if you can see my screen. And I'll start.
We can see your screen, but it's in the presenter mode. So can you please go to the presentation mode?
Okay. I'm not sure. I think let me go back. 1 second.
Okay. Yes, it's perfect now. Thank you.
Okay. Sorry. I want to go back to my beginning. Let me go to that. Okay. So as I mentioned, and as was mentioned, the title of this talk is Gene Therapy, the Era of the Hematopoietic Stem Cell. And so I will be talking today about what is a hematopoietic stem cell, HSC for short and easier to pronounce. What are the current treatments available, and how it works, and what the challenges are with our current way of working in terms of HCC. And also gene therapy, what is it? How does it work? What are the challenges? What are the prospects? And what is the future in this area? So hematopoietic stem cells are an immature cell that is located in the red blood marrow and the peripheral blood. It has two very unique qualities, the first of which is that it can replicate itself to form more itself over and over again. So this is a property that is used in the laboratory to generate lots and lots and create lots of hematopoietic stem cells. The other interesting thing about HSCs is that they can differentiate it into all types of blood cells. And this process is called hematopoiesis. Sorry, it's quite a mouthful. So I mentioned before that it can differentiate into many different types of cells. And these are the number of cells that it can differentiate into. So it differentiates into platelets, to neutrophils, to red blood cells, to monocytes, B cells, and T cells. And why that's particularly useful is that different diseases have different genetic mutations, and these are causing these diseases, which if we can replace these problematic gene mutations, we can essentially cure the disease. So I've listed here a number of different conditions. Hemophilia, a lot of you will know, is the inability for the blood to clot. And you can see that the platelets are often affected in this way by, in the case of hemophilia. And this condition here is called Wiskott-Aldrich syndrome, and it's an actual disease that I'm actually working with. And it essentially means that the patients have less ability for the blood to clot, and so they do have severe bleeding, which can be fatal or detrimental. And as we go along, we can see all the different diseases and the different type of cells that are impacted by these genes that are faulty. So what I should also mention is that adult stem cells don't have the ability to differentiate, and they are very rare. So this is why we're utilizing the properties of hematopoietic stem cells, which are the immature cells. Currently, for some of the rare diseases that were on the previous slide, the only way to treat them is using a donor match who is similar but not the same as the recipient. And how this works is that the healthy donor will have his blood cells, stem cells, harvested, and these blown cells will then be stored until the time they will be injected into the transplant recipient. Prior to these cells being transfused into the patient itself will have to go through chemotherapy or radiation treatment. And this process is really quite a lengthy process, but equally is quite a serious and difficult process as well. And there are complications which will occur because of the allogenic hematopoietic stem cell transplantation. Some of the aggressive procedures I mentioned, it can create short and long-term risks, as well as compromise the immuno-mediated complications as well. Some of the risks include fertility problems, early menopause, increased risk of developing infections, autoimmune conditions, as well as cancers. And we have the added burden that hematopoietic stem cell transplants use donor cells, and there's a risk of rejection and subsequent graft versus host disease and death. And especially in cases where the match is not a sibling, we have a lot more of rejections or graft host disease conditions. So the alternative is what we call gene therapy, utilizing the same cells that we use in allogenic transplantation. So what happens here is that there is a mutation in the gene that is identified as causing that particular disease. So the first step is identifying that disease, and then it is a case of isolating target cells from the hematopoietic stem cells from the patient, which has the faulty genes. And then utilizing a vehicle to deliver the identified gene, the normal identified gene or the therapeutic gene into the patient. And that is done by taking the target cells, the isolated cells from the patient, assembling the correct gene on a vehicle, and then these genetically modified cells will then be injected back into the patient, and these will be expressed in the patient, and therefore the patient will essentially have a therapeutic and disease cure. It all looks very simplistic when I talk about it on paper, but the reality is this process has many steps, and this needs to be considered when we're actually looking at how we do gene therapy. So I mentioned that the cells are taken from the individual, so they're collected from the bone marrow and the peripheral blood, and they are also, at the same time as taking the cells, the physicians will have a backup dose in case of for any reason these cells do not transfuse or transinduce into the patient. And so they will also then take the desired cells, in this case CD334 plus positive cells, and they will then modify it ex vivo, so that's outside of the host. In the meantime, the patient will have to undergo conditioning, very similar to what happens in allogenic therapy, and they will undergo a course of chemotherapy, which will deplete all the endogenous HSC cells from the bone marrow and make space for these new ex vivo cells. Once these modified cells have been prepared, they will then be reinfused back into the patient, and the cells then will engraft in the bone marrow, and the cells then will self-renew, potentially for the lifetime, giving rise to differential progeny along all hemopoietic lineages. So all those cells that I mentioned earlier will then now be have the correct gene in them to ensure that the patient doesn't have the disease. I did mention that there is a vehicle which we use to carry the gene, with the corrected gene back into the patient, and these are called vectors. This is one way of doing this. There are other ways, but this is a viral vector, and it relies on virus. The virus is, we've taken out the active component of the virus and reinserting the correct genetic sequence, in this case, in order to ensure that the patient receives the corrected genes, and then this will be re-injected into the subject, to the patient. There are certain requirements for genetic therapy which mean success. So I did mention that we insert a therapeutic gene into the DNA, or we can edit genes ex vivo and put the corrected sequence by the same method. So this has to be correct, but equally we have to ensure that we have to monitor and ensure that the cells are replicating, and also that the cells are self-renewing, and also that they are differentiating into the different lineages. So as I mentioned, there's a lot of places where this could go wrong. The most commonly used gene transfer vehicle I mentioned is the retrovirus, and that's been used quite extensively in gene therapy. However, as I talk later on, there are reasons why we are moving more towards a different type of vector called a lenti vector. So these are important parts in order to get successful gene therapy. Here I mention all the different applications of cell and gene therapy, and as you can see, in the future there is lots of scope for development of new therapies for some of our common diseases, as well as more rare diseases. So there's lung disorders, hepatic disorders, diabetes, cardiovascular diseases, skin and eye conditions, brain condition disorders, and then more vascular disorders, as well as more embryonic developments as well. So the idea of hematopoietic stem cells and the use of those in treating disease could be extended to many, many, many areas going forward. This was taken from a publication in Helio, which showed that in terms of active diseases, clinical trials in regenerative medicines and using advanced cell and gene therapies, there are a lot of trials active at the moment. Over 300 trials in phase 1, over 600 in phase 2, and a good number in phase 3. So that's really promising to see. Also, in terms of conditions, we are seeing a lot more trials in oncology, and so this is really encouraging, because we so far been focusing a lot more on the rarer disease, and in terms of neurodegenerative disorders, 94 trials is quite significant, as well as 73 trials in infectious diseases. There are challenges, as I mentioned, with gene therapy and in terms of hematopoietic stem cells. The first of these is that these are very costly treatments. As you can see, the number of processes that have to be got right and have to be precise. We only have one chance really to do this, maybe backup, but generally we only have one chance. So it is extremely costly to manufacture, to treat. It requires a lot of, it has big impacts on the family, on in lots of cases, these normal children are coming from a different country and have to stay a significant amount of time within another country to have this treatment. So there's a lot of costs, and this is something that is a real challenge in the industry at the moment. Also, we want to make this treatment available to all, so, and also have sustainability and a fair public access for all. We don't want it to be a therapy that only those who can afford. As I mentioned, travel is a really big thing to another country where you don't speak the language, or where you have to be away from the rest of your family. We know that this is another challenge. And I have mentioned briefly about the complexity of product manufacturing. This is a highly personalized therapy. We're taking someone's individual genes and we are processing them. So this is another complexity that we hope to improve on as time goes on. There is a need to reward industry investors in this area. It is a complex, and also it is an expensive treatment, and industries are putting a lot of money into it. And at the moment, there is little financial reward because obviously you have to do all the groundwork. So the immediate short-term goals, short-term benefits and goals are not necessarily obvious. But we do need more investment, and obviously we need to reward those who invest in this technology. In the case of the gene therapy using the stem cells, I did mention about the vectors and this processing, and also the harvesting of the cells once they're back inside the patient, and the transduction process. These all need to be improved, and there are I know trials and other experiments going on to improve these as well. And I will mention some of the cons at the moment. In one such publication, I noted that there was a delay in one of the trials because of emergence of leukemia, in a fraction of patients treated via gene therapy. And this was related, it was found, to the vector, and I talked about the viral vector. We often use retroviruses, which have an ability to have a gene next to it that will cause cancers. So this is why a lot of companies are moving towards lentiviruses and lentivectors. And so the other issue I mentioned is about the low efficiency of the gene transfer and the risk of genotoxicity. This also has been shown to be the case with some of the early retroviral vectors used in the trials. However, we are making great improvements using lentiviruses. That all said, a company most recently in February 21 has had to stop its clinical studies on a promising gene therapy for the blood disorder sickle cell anemia after 2 of its patients developed leukemias like cancers. And so although there are these incidents, that also should be mentioned that there is a lot of success as well. I'm working for a company at the moment working in the Wascott-Aldrich syndrome, and you know, there are children who have diseases where they have been seeing improvements in their health in terms of, you know, the condition, but equally, you know, at an age now where you can almost see that there is a real impact from having the gene therapy. Here is a picture of, I don't know if everyone knows, but this is sickle cell anemia. This is very close to my heart. I do carry the sickle cell trait, but I also have a nephew who has the full-blown disease. So it would be really nice to see the conditions such as sickle cell anemia having a treatment option for patients available. In terms of the future of clinical trials using HSC-GT, there is increased confidence with safety and therapeutic potential of this therapy based on the studies that have been conducted at the moment. there is a 15-year follow-up period for gene therapies, and which is why at this time we don't consider any therapies to be a cure until the 15-year mark. Also, I mentioned about the clinical testing of lentivirus vectors. That has moved gene therapy using HSCs into a route, a different route in terms of treating some of these lethal conditions. And so now HSC GT is considered as a treatment option, and patient inclusion criteria are expanding based primarily on the lack of suitable match HSC donors, and equally some of the risks that I mentioned before about allogenic transplantation are the reasons why this method seems to be a lot more trials are popping up because of that. And something to note, as I mentioned earlier, the emergence of trials in HCC for cancers is really growing because of the potential biotherapeutics that can be achieved from it, but equally because of the safety profile that's been shown. And we will hope in the future that there will be applications, more expanded applications of this therapy to replace or to edit faulty inherited genes, and also to engineer new products based on the properties of the hematopoietic stem cells. Some very good news in terms of cell and gene therapy is that in 2020, the FDA approved the first CAR-T cell therapy for mantle cell lymphoma from Kite Pharma and Gilead. And Europe and Japan and Canada approved US approved it in 2019, a treatment for spinal muscular atrophy in children. And that's by Novartis Gene Therapy Group. And also in Canada, there was approval of a treatment for retinal dystrophy. So again, and by Spark Therapeutics. So again, a really good idea of what work has been going on and what the regulators are saying about gene therapy and cell therapy. And here is just a list of the different conditions and genetic diseases that are being treated via allogenic transplantation, as well as retrovirus gene therapy and lentiviral gene therapy. And you can see here that there's a number of diseases in the primary immune deficiencies that have been treated in this way. And we hope that this will grow in terms of the number of different conditions that have been treated in this way. Many of these are rare diseases, and this was the disease I mentioned that I am currently working with the company on. Also, in terms of the other categories of diseases, we're seeing hemoglobin pathologies like sickle cell anemia, which affects some of our minority groups. Also, beta thalassemia also affects the minority group as well. So we are starting to see some of these diseases being treated in this way as well. And also storage and metabolic disorders, as well as congenital cytopenias and stem cell defects. So it's really nice to see a larger number of diseases being treated in this way. So the prospects and the future of hematopoietic stem cell gene therapy is that it may eventually surpass allogenic HSCT because of the lower treatment related short and long-term morbidity, and also in some cases improved efficacy. HSCT-GT exploits the autologous cells as opposed to the allogenic cells, and therefore is virtually available to every patient. So we've all got our own cells, and no risk of graft versus host disease, and that's very significant. And also, in most cases, it does not impact on the immunological barriers in the recipient because it's cell. and it allows the exploitation of a range of reduced intense conditionings. I mentioned that the patient has to go through conditioning, quite severe condition of chemotherapy. We believe that the conditioning may be less intense. The conditioning regimens are based on the allogenic HSCT transplantation, and we may not need as much intense conditioning as that in order for the reintroduction of the modified cells. And new depleting drugs are being researched and tested to get a better target hematopoietic progenitor, while sparing the stromal components of bone marrow. So just taking out the part of the bone marrow that's actually needed. These are researchers going into that as well. And so that will have less impact. And if we use these strategies, what the hope is that we'll be able to alleviate the long hospitalizations. I did mention that sometimes a patient is in isolation, obviously during the chemotherapy piece, for up to a month, and the treatment at the hospital may be 3 months. They may be there for 3 months. It's very impactful on people and families. So the hope is that eventually we'll get to the point where the gene therapy can be done and patients can be outpatients as well. And you know, we too talk about this as a therapy, but actually it could be a robust cure. It maybe is too early days to talk about cures, but it certainly is a one-off treatment that would alter completely the genetic the makeup of cells. So we hope that this way we will dramatically improve survival and quality of life. And as I mentioned, this is a lifetime treatment. And in the long run, it would save on conventional symptomatic treatments, or visits to hospital will be less frequent. Medicines people won't need as much medicines as well. So the dawn of a new era, for sure. HSCT-GT challenges the traditional framework of drug development and requires creative solutions to grant sustainability, commercialization, and fair public access. HSCT-GT may be a way to address the issue of diversity, since it's tailored to the individual's DNA and makeup, and it offers the reward of dramatic therapeutic benefit. We need to continue to form strong relationships between all stakeholders in science, medicine, pharma industry, the advocacy groups, the regulators, and the decision makers across all nations. It's grounded on scientific evidence and inspired by the ethical principles of a just society, which will enable addressing challenges and capturing the promise of providing long-awaited relief to some of the unmet disease burdens on our humankind. So this is really the reason why I feel that this is really strongly about this approach, because the hope is to eradicate some of these diseases where there is an unmet need. And I think that's my last slide. I'd like to thank you for listening, thank this forum for allowing me to speak, and I'm happy to answer any questions.
Thank you very much for a very nice presentation and the good knowledge that we gain of it, especially that gene therapy is now the promising era to solve a lot of problems and like cure a lot of illnesses that till now they are uncurable. For question, we have 2 questions from Mrs. Gail Trelko. First, were the metrics from number of clinical trials based on global open clinical trials? Yes,
they were. Yes. Yes, I can provide the reference as well. So yes, it's based on global.
Thank you very much. And also the other question, do you know how long after the gene therapy infusion that the 2 patients in the Bluebird bio trials developed the leukemia-like carcinoma? And what period follow-up were the 2 patients?
Yeah, I can't remember the exact time frame between them developing the carcinoma. I've got a feeling it might be about a year, but I'd have to check that. And these patients are followed up for 15 years in any case. So they will be regularly followed up with the trials. Although the trial has stopped recruiting, those patients will have ongoing follow-up throughout, and obviously treatment for their leukemias. I think that the main concern about some of these, I mentioned the viral vectors, is that they can cause some kind of oncogene process, and that's why a lot of companies are trying to work on the vector to make sure, or on it we have promoters as well in there. So I think that these are the reasons why that they think there's a link between that and the leukemias that were created.
Yeah, thank you for your answer. I have like a comment following up your answer. We know that the viruses that have been used as a vectors for gene therapy have some side effects. And now there are growing interest of using like nanovectors, nanotechnology-based molecules. For example, in patisiran, which is an FDA-approved single interfering RNA, small interfering RNA, and also with the vaccine, COVID-19 vaccine now, it's mRNA and with the lipid nanocapsule, lipid nanoparticles. So do you think that nanotechnology and nanomedicine could be an alternative solution? And will this be helpful in preventing those side effects, or you doubt it?
I do agree with you. I'd have to look into it more, but I definitely think nanotechnology could be definitely useful here. Absolutely. And I would welcome anyone who's doing research on that to bring that forward, because I think this is definitely something that could alleviate some of the side effects from the therapy. Yes.
Yeah, thank you very much. And also another question, we know whenever we go to gene therapy and cell therapy, we have the ethical problems, and the ethical community, they are going to say, okay, this is good, this is nice, this is not. Do you think that we have now regulations that are helpful for gene therapy to go forward, or we are still tackled with those problems?
No, the regulators, there's the advanced medical therapies, the ADMTP, are very aware of these, the regulations. The regulations are strictly strict around gene therapy, and rightly so. They, we will be inspected for every single trial is inspected, but I think the regulators, as well as the rest of the world, are have bought in to the fact of that this is a way forward in terms of healthcare, and in terms of healthcare for all conditions. And in terms of ethics, obviously we have to get to ethics approval for all these trials as well, and they are bought into it as well. There will always be the element where people think that we are muddling with what we're born with. However, if you suffer from a rare condition, and if you see some of these children whose life is short because of their condition, you would understand why we need to do this kind of therapy. But it shouldn't be underestimated how closely it must be regulated, because we don't want to have any doing it for designer reasons, to have the right makeup. So to speak, this is really about treating unmet need and treating diseases that are currently not treated effectively at all.
And final concern for me about this, because for me, technically, personally, it's a very interesting field. So how do you feel patients are going along with participating in clinical trials for gene therapy? Because till now we have patients who have the fear, like gene therapy might interfere with our DNA, chromosomes, we will have genetic disorders in the future. So is people going more friendly with those techniques, or they still have those fears?
I think it's a mixture. I think a lot of the diseases I mentioned there affect a lot of children, young children. So the decisions are being made by parents. And I've been lucky enough to speak, to hear some parents from advocacy groups talking about their child's life when they were 2 and the limitations of their life. And those kind of parents are looking for a cure. And often these diseases, you've got to remember, nobody knows what they are. So there's no screening at birth, for example, to know what these, and there's no biological markers to understand what these diseases are. So lots of the time, these diseases are not being found until very late. So for those kind of parents, when they find that there is a potential cure, it seems very straightforward. But for people who are not experiencing those diseases, there will be a lot more skepticism. And this is why education by the doctors and the healthcare professionals and the industry as a whole presenting this, very simplistically to everybody, I think would help people to buy more into the gene therapy. I do take people's comments about it being scared of gene therapy. As soon as you talk about RNA, DNA changing, people start to get very worried. But I do think it's becoming a lot more established. We are seeing trials, 15-year trials. I think at that point, people will, be more bought into it because they actually can see the safety profiles of a lot of these therapies. And I think it will become the thing of the future, most definitely.
Thank you very much for that very nice presentation and for the very precious information you gave us. And thank you for shedding the light for this very interesting topic for the future.
Thank you. Thank you very much, and thanks to the audience.
You're most welcome, and we are very happy to have you in